Thank you very much. It's really my pleasure to have the chance to speak with you today. Meetings like this are really great. It really feels like a gathering of old friends as well as a chance to meet new people. So much like Sajeevan, I've been working with Paul for quite some time. For those of you who don't know me, I actually started my master's thesis with Paul 19 years ago. Then I managed in the short term to escape his gravitational pull. I flew across the pond to do my PhD at Imperial College London in England, but then I couldn't resist the temptation to come back and be part of this really great project, and I joined the faculty here in 2009. So I've worked in a variety of study systems with a heavy focus on Arctic biodiversity and freshwater systems, and as well in our group we use a lot of public data generated and released by many people in this room, so thank you. So I'm an evolutionary biologist. My interests include molecular evolution, macroevolution, and patterns of diversification. And recently, my interests in bioinformatics have been increasing. In particular, I'm interested in using some of the knowledge we've gained about evolutionary processes and evolutionary patterns in order to inform the creation of bioinformatics tools that serve a broader community. So it's my pleasure to talk briefly about a new project that we're just launching now. This is a highly collaborative project involving many people here at the Center for Biodiversity Genomics. As well, we're starting to ramp up recruitment of students who are associated with this project. And there are as well more collaborators joining from across campus, including from the Department of Math and Statistics, as well as from School of Computer Science. So I think it's really great to see a larger diversity of brains starting to turn attention to biodiversity research and problems in this area. So as I mentioned, this is a brand new project. Hopefully the formal funding announcement will be soon, but it's a 3-year project just starting this October with a multidisciplinary team working together on this. And our overarching goal is to create bioinformatics tools that help to facilitate high-throughput biodiversity science in service of the iBOL community as well as the broader scientific community. So as we've seen yesterday, and as many of you know, now there's capacity to take a set of sequences and draw some conclusions from it. So such as, for example, from a Malaise trap sample. For example, we could say, in a Malaise trap, there are 512 bins in this sample. But I think members of this community have pointed out that we want to go beyond this, right? You want to know something about what's in that sample. What's the taxonomic composition of samples and the relative composition among samples? And I think that there's a need to go beyond that as well towards functional annotation of samples. So not just who's in it, but what are they doing in the community? So for example, it would be very helpful information to know at a particular site your relative proportion of reeds from herbivores is increasing. Or maybe, hey, you've got a blood feeder in your sample that's never been recorded in your country before. Maybe you want to take another look at that sample in more detail. So those types of functional annotations, I think, would be really helpful to serve the broader community. So together, we've sort of broken this larger task down to a few much more specific projects, tackling some issues relating to denoising, clustering, taxon assignment, functional assignment, leading into increased capacity for global biodiversity surveillance across space and across time, considering both taxonomic and functional components of diversity. So we'll just briefly speak the most, I guess, about Project 1, and then just very briefly outline the goals of the other projects to work on over the next couple of years. So the first target is to improve denoising capacity with emphasis on protein coding markers. So we know quite a lot about some of the core protein coding markers in terms of their patterns of amino acid variability among positions, for example. So we know quite a lot about these markers, and that hasn't really fully been capitalized upon yet in terms of building bioinformatics tools. So one thing we know about high-throughput sequencers is that in general, they tend to have a higher error rate than Sanger. Now, Evgeny mentioned excellent success with SQL. That's based on building consensus sequences. But the capacity of a variety of types of sequencers to contribute to biodiversity science is very high, including ion torrent, Illumina, MinION, and various platforms. And the error rates tend to be high enough that they can even generate erroneous operational taxonomic units. And various researchers deal with this in different ways, often by setting stringent filtering thresholds, for example, throwing out sequences that have few representatives. And there have been protocols developed as well for filtering out OTUs, or operational taxonomic units, that show unusual coexistence patterns with others. And I think these have been necessary, but the unfortunate thing is you can also miss real OTUs by that process. And we may want to detect rare OTUs. What if we're, for example, monitoring for invasive species, and we want to be able to detect at very low read abundance an invasive species in an environmental DNA sample? Or what if our target is environmental monitoring for endangered species? Is there a way that we can improve the data so that we don't have to throw out those rarer reads? So in the interest of time, I won't go too much into the details, but on this particular project, we've started with a smaller scale pilot project and focusing in first on the SQL platform because it's being ramped up so much, but then also plan to expand to a variety of other sequencers as well. In this particular case, we're starting at the very front end, looking at read errors in the tags. And the goal of the project, of this first step of the project, is to determine the types of errors that the sequencer makes so that the tags can be designed to be robust to the type of errors that the sequencer makes. So again, this is very preliminary results based on about 250,000 sequences. So just to give you an idea of what we're trying to go for. So here the row labels are the tag bases, ATCG, and on the column labels are what the read was. And along the diagonal, you can see that in general, there's a 92 to 96% correspondence. But there are also some errors. So just as one example, about 4% of A's were erroneously called as a deletion. Those bases were missing. So we're interested in fitting these types of error profiles in more detail using more data, and then as well trying to predict when errors occur. So for example, does it matter what's slightly upstream or downstream of that position? Does that help us to predict when there might be an error? As well, it's important to keep in mind that indels are a really important source of error as well, and so our preliminary results suggest a little bit more propensity for example, 2 base pair insertions rather than 2 base pair deletions. And this kind of data can be helpful for building up a denoising algorithm. So again, this is at the very beginning. This project just started officially 2 weeks ago. So we'll be hopefully discussing more results at the next conference and beyond. But the anticipated outcomes of this research would include a better understanding of error rates and error types using larger data sets, contributing to the front-end design of projects in terms of design of tags to be robust to the types of errors that the sequencers make, as well use information about patterns of variability in sequences and amino acids to detect and correct sequencing errors. And as well, I think there would be really a need to build not only error models for specific sequencers, but to build an adaptable pipeline that builds a denoiser tool. Because I think it's really helpful for our community if we can be responsive to technological advances. A new MinION model just came out recently, for example, which I think could have great utility at point of need, such as at port settings or as well in remote field settings. So the idea here would be also to move towards building tools that can create a denoiser tool on a faster time frame. So in the other projects, I'll just briefly outline the goals of the project. So in the microbiome literature and beyond, there's been movement towards various ways of looking at biodiversity, including looking at amplicon sequence variation, looking at phylogenetic diversity. So I am interested in exploring those as well. But for our community, I do still think that the species unit has a special place in the study of biological diversity from both a scientific perspective and also from the perspective of linking with policy. So policy, for example, about invasive species, agriculturist pests, etc., are often based around the species unit. So I still see an important place for clustering and, for example, the bin algorithm as being a really important contributor to the iBOL 2 project. However, it can be a little bit complex. For example, Torber and Ekram and Elizabeth Stewart have pointed out that they tend to find high intraspecific variability in their group, which is a group of freshwater insects, very important for biomonitoring. So in addition to taxonomic variability, there's also variation in sequence length and sequence position among studies. And in general, we see that studies in using high-throughput sequencing may be more heterogeneous than we were used to in iBOL 1, where standard primers were usually used. So there's been a great increase in studies of diet, for example, studies of environmental DNA, or other conditions where, or museum specimens where DNA may be degraded. So we have variability along the barcode marker in terms of where these shorter sequences lie. And so we see variation in sequence length as well as position. And sometimes those may fall in higher variability regions of our marker genes, or sometimes in lower variability regions. So this project will be about trying to optimize the parameters for dealing with OTUs or for clustering OTUs under these varying conditions. So Jeevan did mention a higher taxonomic classifier, which is really important in general for automating the process of processing large-scale sequences. So this particular project will also make a contribution towards that goal in terms of bringing in some machine learning techniques to try to understand if there are sequence features that are maybe even more helpful than ones we've previously thought about. So certainly we'll be looking at simpler sequence features like amino acid composition, but also seeing if there are any other features, such as the ordering of features along the sequence, that may be really helpful for assigning sequences to their place in the taxonomic and phylogenetic hierarchy. So then we come to functional annotation, which I think is a really interesting area with a lot of potential. So we're planning to explore a few different approaches, some that are dependent on taxonomy, where if a sequence gets, say, a family level ID, then the functional annotation to a certain level would follow that taxonomic identification. As well, it may be possible, or at least interesting, to explore if some types of functional annotations could be made independent of taxonomy on the basis of sequence features. As well, we're starting some work on phylogenetically informed trait imputation to see if we can accurately predict some types of traits in cases when the data are missing. And I think that this is promising, at least for high-level functional traits, because some traits such as predaceous versus herbivorous lifestyle don't sort of vary willy-nilly on the tips of the taxonomic or the tips of a phylogenetic tree. They tend to be conserved. So for example, sometimes an entire family or subfamily of insect might share a same trait, such as a predaceous feeding mode, for example. So I think, at least at high-level functional traits, that some of these approaches may be very beneficial in terms of helping with functional annotation. And if the methods that turn out to be the most successful are more computationally expensive than we might want, we could consider a system of annotation at the bin level, for example, and of course that would need some likelihood or probability values around it. So this is my last slide, just to let you know that the end goal would be to, of course, publish the tools and share them, but as well, the tools that are most useful would get incorporated into BOLD and mBrave to be made more broadly available for the community. And so one of the reasons we wanted to present this at such an early stage is to open the door for feedback and discussion. So if you have suggestions about these lines of research or requests for functionality that you think would be really helpful, it'd be great to chat with you early on in the program so that there's a chance to incorporate your input. Okay, so thank you very much.